Dear students, today I am going to speak on the subviral particles which supposedly causes diseases in plants and animals. Before the invention of bacterial filter, it was believed that the bacteria are the smallest organism which causes disease in the plants and animals. But after passing the bacteria through filter and the bacteria they remained on the surface of bacterial filter, despite that the filtrate had the capacity to cause disease in the healthier ones. This made to believe the scientists that there are some particles which are smaller than bacteria which causes diseases, and this led to the discovery of viruses. But these subviral particles, they are even smaller than viruses, and even then they have the capacity to cause the diseases in plants and animals. And today we are going to discuss some of the subviral particles which are infectious in nature. First of all, we'll take up viroids. I need not to tell you that these are the subviral particles. Viroids were discovered by Diner and Raymer, and the first viroid which was discovered was potato spindle tuber viroid, and it is also called as PSTV. This potato spindle tuber viroid causes disease in potato. It was smaller than viruses and devoid of protein coat. And you might have studied, or you in the previous lecture, I spoke regarding the viruses which contains envelope of proteins called as capsid, which is made up of capsomere, and the genetic material is encased in the capsid. But here in viroids, they are devoid of any kind of protein coat, and they are made up of RNA alone and nothing else. They do not code for any protein but are merely small fragments of RNA of low molecular weights and replicates autonomously whenever they infect a organism. These viroids actually are restricted to plants. That means they do not cause disease to animals reportedly restricted to only to plants. And we haven't discovered any disease caused by viroids in the animals. The example is velvet tobacco mosaic virus. This is also a viroid, not a full-fledged virus, and the viroid genome is about 1.1 to 10 to the power of 5 to 1.3 into 10 raised to power 5 Dalton molecular weight nucleic acid, and the nucleic acid is RNA, not DNA. It tends to remain, the nucleic acid I am talking about, it tends to remain circular in a circular form and is naked. That means it is not enveloped in any kind of proteins. The number of nucleotides contained by the simple naked RNA of viroids are around 250 to 350 nucleotides long. They lack the initiation codon AUG, and due to complementary sequences present in the RNA strand, they tend to form double-stranded region due to pairing up. You can see how it forms a double strand. This portion on the leftmost part is the terminal domain. Then following terminal domain is pathogenicity domain, and after pathogenicity domain you can see in the picture or slide that it has central conserved region. The conserved region means these are remain undisturbed during the course of evolution. Following central conserved region domain, there is a variable domain, and at the rightmost part you can see it is a rigid terminal domain. So this is the simple RNA molecule which is folded upon itself, and uh, then uh, then comes another let us talk about the origin of viroids, that how do such subviral particles they get originated. Watson in 1987 said that these viroids are of cellular origin. That means they haven't originated from viruses and their genome, because what made him this conclusion is they did not induce the biosynthetic machinery of their host for their own replication. This was the reason cited by Watson. Then Reisner and Gross said that they are actually the degenerate virus entities. That means they haven't originated from the cells. Diener, another scientist, said that they have originated from the introns, and the viroids might originated from the spliced and circularized introns. You might have studied, I don't know whether it is known to you or not, that the introns are spliced during the splicing of genes, and the exons they get ligated. Exons are actually the coding parts which codes for proteins, whereas introns are they get spliced because they are of no use. On circularization of these introns, they escape the degradation process which is normally followed when the introns are removed from the genes. When they escape degradation, such introns acquire appropriate recognition sequences. They might be transcribed by the host enzymes which are capable of functioning as RNA polymerase. That means that they escape the control mechanism of host cell and they start their own replication and translation procedures. And then how the viroids they replicate by RNA dependent DNA polymerases, which is present to a limited extent in normal plant cells. Another probable method is through use of reverse transcriptase. The viroid RNA upon infection uses reverse transcriptase and forms a DNA. This DNA then after transcription forms viroid RNA. So this is about the viroids, which are only made up of RNA molecules, and they are not they do not cause any disease in animals reportedly, but do cause diseases in plants. Now come to another subviral particles which are virusoids. The first one was viroids. The second one is virusoids. It was discovered by J.W. Randles, and these are small circular RNA molecules similar to viroids. Let me repeat that the viroids are also made up of RNA, and that too of low molecular weight. The virusoids all are also made up of small circular RNA, but are always linked with large molecules of viral RNA. That means some component of RNA of this subcellular subviral particle is drawn from the virus. That is, they are part of viral genome and are essential for replication of large viral RNA. That I am talking about the part of RNA which is of viral origin appended to the small circular RNA. The small satellite RNA molecules associated with the virus is capable of replication in a virus infected host cell. It plays only in the cells which are infected by the viruses, otherwise immaterial. Then uh, then we have third subviral particle, which is prions. These are also called as proteinaceous infectious particles and were first discovered by Stanley and Prusiner in 1997. These PRP proteins, they are hydrophobic in nature and they are 100 times smaller than viruses and composed of proteins only. That means they don't have any kind of nucleic acid associated with their name. And these proteins are called as PRP proteins or prion proteins. And as I've told you, they are hydrophobic in nature and about 33 to 35 kilodaltons. The electron microscopic studies about them reveal that the 1000 PRP proteins, about 1000 PRP proteins, they aggregate to form prion rods, which assumes the proportion of 10 to 20 nanometer diameter and 100 to 200 nanometer length. This is their infectious or virulent morph or form. These are believed to spread by many vertebrates which are responsible for the spread of these diseases. Now I would like to mention the diseases which are caused by prions. The first one is scrapie disease, which is a neurological disease of sheep and goat. Another disease which is caused in the cattle is mad cow disease. It is also named as bovine spongiform encephalopathy. It is also a neurological order of cattle, and it was reached to epidemic proportion in the United Kingdom because their cattle was fed with bone meat made up from cattle itself. Then we have third disease which is caused by prions is CJD, which is Creutzfeldt-Jakob disease. The third one is Gerstmann Straussler Skinner Schinker disease. It is also called as GSS. Then we have Kuru disease, which is the disease caused in human beings. It invokes involuntary tremors, dementia, and it was first reported in Kuru tribe of New Guinea because they had a ritual. They practiced a ritual which was cannibalism, which involved the eating of the brain part of brains of their deceased near ones as a mark of homage and respect to them. When it was discovered that it is caused because of this cannibalism practice, it was done away with, and after this cannibalism ritual, was eliminated, and the disease also got eliminated from Kuru tribe. Another disease which is caused due to prions is fetal insomnia, which is also caused in human beings, and it is homologous to this mad cow disease, and it is a disease of human gene. So this is all in this lecture, dear students, and this is thank you so much